research. And today we have a very special guest from Columbia University, Rando Allikmets. I was trying to Google this morning what Rando means, and I found only one translation or meaning of the name, which means king in one of the Germanic languages. So it really fits very well to Rando. Rando King Allikmets. That makes perfect sense to me. So it's a wonderful that we have really large number of great scientists in our community. And it's also very pleasant to remember each of those wonderful scientists for something that they did very meaningful in vision research, that open up a new area of research, open up new ideas and a lot of work that follow up on the discovery. And Rando clearly has that golden stone that changed the way how we look at a very major disease, Stargardt disease, with his discovery of gene responsible for that. And that's how I'm going to always remember Rando on his incredible paper on the sequencing identification of this gene and association with Stargardt's disease. So Rando, you have that golden stone, and we're very delighted to have you here. And Emily.
Hi, everybody. Thank you for joining us for our seminar this morning. My name is Emily Tom, and I am a fourth-year MD PhD student in Dorota Skaranska Kravchuk's lab. It is my great pleasure to introduce today's distinguished speaker, Dr. Rando Allikmets, William and Donna Aquavella Professor in Ophthalmology and Pathology and Cell Biology, and the Director of the Laboratory of Molecular Genetics at Columbia University in New York. Dr. Allikmets earned his PhD in molecular biology from the Institute of Bioorganic Chemistry in Moscow and completed a postdoctoral fellowship at the Karolinska Institute in Sweden, followed by a second postdoctoral fellowship at the National Cancer Institute. Prior to entering the field of genetics of eye diseases, Dr. Allikmets' interests were primarily in the fields of human genome and cancer research. He cloned and characterized over 30 genes in the ATP binding cassette, or ABC, transporter superfamily, including several genes involved in human inherited diseases. He then determined that one of these genes, ABCA4, was causative for Stargardt disease and later characterized it as the first gene associated with age-related macular degeneration. Currently, Dr. Allikmets' research interests include developing gene and small molecule therapies for retinal disorders, genetic studies of Mendelian and complex eye diseases, and microarray-based, next-gen sequencing-based diagnostic applications for ophthalmic conditions. He has made tremendous contributions to our understanding of the role of genetics in several eye diseases. He was awarded the 2018 Ludwig von Salman Prize for his efforts in greatly increasing both the basic and clinical understanding of vision in ophthalmology. He has published over 200 original peer-reviewed articles and holds several patents for his innovative technologies. It is truly our pleasure to welcome you here today. Please give a warm welcome to Dr. Rando Allikmets, who will be presenting his talk entitled Precision Ophthalmology for Stargardt ABCA4 disease.
Hey, thank you very much. It's very great to see you all. Let me see if I can get my screen shared. You can see the screen well?
Yes.
Okay, so I'm really honored to be invited to your seminars, because I know that these have been going on a while, and when I accepted the invitation,
Rando, we lost, you muted and we lost your presentation.
Yeah, it for some reason it said that my Zoom quit, you know. So this is Colombia, so there is nothing. You see it again, I guess.
You're back. Oh well.
Okay. Hopefully it will last no more than 5 seconds. What I was saying is that only yesterday I realized that my seminar is how it says again, the Zoom quit. I don't know. We'll see.
It's working.
Is on Friday the 13th. I don't know if that I should make something of it or, but that's a fact. So again, thank you, Chris and Emily, for the nice introduction. Chris and I, we go back quite a while. We never actually collaborated, but we always discussed science because there are only 2 biochemists I ask advice. One is Chris and the other is Bob Molday, who is also online if I have any questions, since I understand nothing in biochemistry. And I always said that I'm the only one that can argue with Chris because we are in the same weight class, and now we are even closer related because we probably have to pick up machine guns and go to defend our fatherlands sometime soon. So I changed my title of the talk a little bit because, again, to give an overview of what's happening in Stargardt disease research in ABCA4 disease research, it's very difficult to pick, you know, some specific things. I have been giving more of a clinically related presentations, but in this time I decided to put some more science into it that reminded me of an incident a few years ago when we published a paper or submitted a paper, then got back the reviewers' comments, and they were quite substantial. And then when we asked, we didn't quite understand what the reviewer meant. The reviewer responded that this reviewer wanted to insert some science into that paper. So I try to do the same today with my talk. So I'll give you the overview of mostly what has happened the last few years, because this field, the field of Stargardt ABCA4 disease, has really developed fast recently. And just a few introductory slides. Stargardt, as it's been historically known, is recessive disease, so it needs both alleles to be mutated. And you'll see it has hugely variable onset and severity. The most well-known things are, you know, the central atrophy and the flex, but you'll see quite soon that this is not always the case. And again, of course, the disease is caused by, it's a single gene disease, it still is. So all the rumors about, you know, being polygenic or so are not true. The protein that is affected is the ABCA4. Here is a slide from Bob Molday's paper. That is the transporter of both 11-cis and all transretinal in the rod and cone outer segment discs. If it is not working, and whether it's absolutely not working or working not that well, what's happening is that a compound called lipofuscin accumulates. That is a major hallmark of a Stargardt disease and always has been considered that, but I'll show you that that's again not always true. But here is a paper that we did together with Janet Sparrow and associates, showing that yes, in Stargardt disease, in most cases, the lipofuscin A to E accumulation is really fast, and that happens at a very early stage of life. Here is, you can see the age-related curve that sometimes Stargardt cases fall into this also. So a good review we published a couple of years ago with my good friend Franz Kremers and Winston from my lab and Rob Collin who has done a lot in terms of therapy of Stargardts. So it's still pretty updated. So if you want to get the deeper look into this, you can go and check it out. So the history is pretty clear. The protein was characterized by Dr. Papermaster very long time ago. And it took, 20 years or so before I somewhat, people say, accidentally cloned the gene, although there was not much of an accident there. My postdoctoral goal that Mike Dean gave me at National Cancer Institute was to clone all human ABC genes. And so if the current postdocs think that they have too large things to deal with or areas, so go and try that, because at that time cloning the genes, mid-90s, was still kind of an art. So we cloned it. We found it being responsible for Stargardt. We associate with AMD. I will not go there today, but if somebody is interested, I can give you the update. But then quite soon, by us and other groups, it was also shown to be a causal gene for many different phenotypes that did not really resemble the classical Stargardt at all. And so it is a complex business. So here, yes, we cloned the gene in 97, so I looked a little bit younger than, but genetically speaking, most of the advances have been coming from my lab. First, we did the microarrays more than 20 years ago. Then we sequenced the entire gene. Then we sequenced the entire locus. That is 140 kb. Then we defined the hypomorphic mutations about 5-6 years ago. That was another major breakthrough. And then now we have been really working on mostly on modifiers, both cis and trans-modifiers. So over the years, so what is it now, 25, 26 years, there are more than 2,000 variants that now cause ABCA4 disease. So again, the disease is really heterogeneous, and all examples here on autofluorescence show cases of ABCA4 disease. And here are just, the short list of all phenotypes that have been associated or caused by mutations, variants in ABCA4, from Stargardt and fundus flying oculatus to cone rod, occult, rapid onset, bullseye, very important subgroup. And even some people claim, including us at the first time, that it was retinitis pigmentosa or RP-like. We haven't published these data, but this is not RP, so we publish it soon. It is still macular degeneration. It's just the end stage looks like RP. So it is a macular, it's a single gene macular disease. And you can see that, and you'll see that a little bit later. I'll jump over it a bit here, that different variants cause very different phenotypes in the progression of ABCA4 disease. So it is clear that genetic or that clinical heterogeneity is determined by genetic variation. And after us and now mostly Franz Kramer's sequencing the entire genomic locus, we found a lot of deep intronic variants, other interesting variants. So right now, these are kind of the percentages of variants in ABCA4, of which by category, about 20% are deleterious, 20% are severe. There is some difference between that. Close to half are so-called moderate, but then there are mild, and then there are very mild that are called hypomorphs. Interestingly, while the ABCA4 locus is very heterogeneous, there are very few large copy number variants there for unknown to me reasons. And again, there have been every now and then attempts to say it's polygenic and dominant, and there are some gender issues. No, that's not the case. So it is really quite classical single gene recessive disease. However, it is not that simple. So again, in terms of where are the mutations, most of them are, of course, in coding regions, but they are also in splice sites deep in tronics, and we're looking now at regulating regions for some missing alleles. Another very important thing to bear in mind is that ABCA4 variants, there are founder variants, not only in racial groups, but even in nationalities. And when I get the results of a patient genetic screening, I can tell pretty much, especially if they are of European descent, where what is their nationality or where they are from, because in every major nation in Europe, of course, now it's more admixed, and in the States it's even more admixed, has their own specific founder variant. And if you look into different racial groups, so it is a totally different picture, whether it's African Americans, whether it's East Asians, not that much South Asians, but I'll come to that. So it is complicated, and other than European populations, the others are not studied that well genetically, so those gaps have to be filled. So again, this shows kind of the old, the first suggestion for a model, that what happens if there are both alleles mutated in ABCA4. This came from my good friend Franz Kremers again, that if you have, two knockout, if you have both knockout variants, you get very severe disease, then called RP. Now it's not. If you have different combinations, you get different expression of the phenotype. Another important point to make here is that about 10% of ABCA4 alleles, and probably higher, are complex, meaning that there are two mutations on the same chromosome. In this case, I don't mean modifiers, I mean two mutations that separately, when they happen, they also cause the disease, although at different severity. So now, this is another difficult point when people ask, so why do you need genetics? Clinicians can easily determine if you have Stargardt or not. So I've shown this thing to clinicians a lot. So as you can see, there are two rows. There are practically identical phenotypes, but if you start looking, genetically, so then the lower row is ABCA4 of different variant combinations. The upper is RDS peripherin or CRX, and I'll touch on that quite soon. So when in early years, when we were screening ABCA4 in cases that were diagnosed clinically with Stargardt, so we always had like 20–25% cases with no mutations when we screened the entire gene. And so then we started figuring out, so what's happening, and from the previous slide, you already guessed that there are so-called phenocopies. So you see here, I mean, this is the study we published a few years ago. Now we have screened several hundred of ABCA4 negative samples and figured out quite a number of them. But here you can see the clinical composition, that about half of them were Stargardt disease clinically characterized, a third of cone pattern, this and that. And we use the collapsing analysis, though this is the method that is successfully used to determine causal or associated genes with complex traits most often, but it can also, of course, be used for a single gene diseases if you have a large enough cohort. In this case, you are not looking at variants separately, but you collapse all the variants in one gene into one data point, and then you compare the frequencies at certain criteria with controls. And you can see here that when we did this exercise, so the first gene, as somewhat expected, was PRPH2 that popped out, and you can see the p-values were even exome-wide significant, that a lot of cases that have been clinically diagnosed with Stargardt actually are RDS peripherin disease. And the opposite is also true. So CRX came out actually quite high because the bullseye phenotype that ZRX cases have is of course mostly associated with Stargardt, but there were some other somewhat surprising findings. Some of these, of course, had to do with how good the clinician was, because it is not that often that RP65 case is diagnosed with Stargardt, but apparently sometimes it is. So here is a slide that shows, specifically the bullseye maculopathy, that when we looked at the ABC for negative and positive cases, so these is the fractions of different genes that caused the bullseye maculopathy. Now the question about, structural and functional studies, and this is a very important question. So you always say that when you find the variant, so the ultimate proof is in functional studies. Now, in ABCA4, it is not that easy, and I'll show you some examples. Some of you know those very well, because some of these we have done in collaboration with Bob Molday and some other people, that we were waiting and to get the structure of ABC4, the fine structure, and it did not happen. But now, with the AlphaFold and especially cryo-EM, several groups have done that. In this case, the examples are from Bob Molday's lab, and I actually suggest reading these two papers to kind of to learn better about, the ABC4 structure and how a variant can influence probably the ABC4 function. Now, but I'll show you on the example of this most famous and most frequent variant in ABC4, the 1961, that things are not that straightforward, and they are still not very clear. So the 1961 is a very famous variant from the very beginning. In cases of European descent, it is in about 20–25% of cases. In, let's say, India, South Asia, it is more than 50%. And of course, this is because of the population frequency of the variant. In Europe, it's about 0.3%. In South Asia, it's 1.5%. And in my early days, Ed Stone tried to discourage everybody to consider this mutation, because in Somalia it is at 10%. Statement was that, of course, it cannot be a disease-causing variant, because otherwise Stargardt will be rampant in Somalia. My answer is that we still don't know. And I did, in my early years, organize an expedition to surrounding Somalia countries and looking at tribes, and it is high there. But now it took us a while to figure out, so what is the reason? So I always say that instead of making a statement based even on your data, you have to figure out why that is the case. So again, the question is, the functional studies suggest that this variant is severe, very severe, and you'll see a couple of slides. Now, genetically and clinically, it is really not severe at all. It is more on the mild side, if we use that term. But it causes specific phenotype, and it is a moderate phenotype. You'll see that. So you'll see that it is mostly causes bullseye, and it practically these patients do not accumulate lipofuscin. So some of them are normal range, some a little elevated, but this is certainly not the major feature in this group. And if you recall, this is about a quarter to a third of Stargardt patients, and there are other patients like that. So this is really shows that the disease process is quite different in different cases and different combinations of mutations. So, okay, so here is a slide again from Bob Molday, and from the very beginning, I think it was first done by Jeremy Nathans and Wei Sun. It was shown that it's very interestingly. So here is the wild type ABCA4, yeah. So, and this, of course, is the AT-based activity in vitro study. If you have the substrate retinal, so the activity really goes up. For other variants, whether there are mutations or not, I will not go there. So what happening is that both the basal activity is lower, and again, the activation is either absent totally or it is also much lower. Now, if you look at the 1961, you can see that adding retinal inhibits actually activity. It doesn't, hinder it. It kind of really brings it down. I've discussed this a lot with Bob Molday. There could be several reasons for that. Technical could be, but this is the only mutation in ABCA4 that acts this way, as far as we know, and that's why there is something funny about it, but it is something that we have to figure out what's happening. Here is another study from Bob Molday's lab showing that the transport activity in vesicles, and again, you can see that 1961 practically has no activity. And final slide from Bob was when you did the transfection of mouse model with mutant ABCA4s and looked at the localization. So you can see that in terms of 1961, about half of them go into outer segments and half of them are maybe inner segments. So again, so these are studies that are done in either in vitro or in certain different environment to the actual in vivo environment in patients. And this you have to bear in mind throughout my talk to the very end. Because what we did, so we made the knock-in mouse for this variant, and we showed that there is absolutely no problem with assembly or trafficking of the protein with this variant. So this is the wild type ABCA4 mouse. This is the homozygous knock-in. You can see that ABCA4 is really where it should be in the outer segments. And this is a het. You can see it's even quantifiable, and of course in knockout you don't see that. So again, so there is no problem to look at. So here are some organoids we made from patients with 1961, and again, of course, these pictures are less pretty than they could be, but you can see that ABCA4 in organoids is also localized to the photoreceptor outer segments as they should be. I mentioned that this variant causes bullseye, and this is pretty much unequivocally so. It goes through this occult dystrophy or optical gap that we called it, where the disease is clearly localized to the fovea and is clearly the photoreceptors die first. And that's again against the common understanding that RP is the first target, or the main target. I think that both options work in Stargardt disease. Again, it depends on mutations. But here you can see that, yeah, you lose foveal cones quickly, and then the center macula, fovea macula collapses, and you have this bullseye phenotype. So this is associated with the vast majority of 1961. Interestingly, the mutation causes always this phenotype. It doesn't matter what's on the other allele. Again, there is a little bit of variation. You'll see that soon. And as I mentioned, 1961 doesn't really accumulate lipofuscin, and probably, you know, this progression explains why. And here, the red dots in the same paper that we had published are 1961 cases, and you can see that very clearly that, you know, the classical disease goes through huge lipofuscin A2 accumulation, but not the 1961 disease. And I'll come back to that, so I'll skip this slide for a bit. Now, what's going on? We don't know. We try to look how this variant is influencing the protein structure. It's not very clear what's going on. There is a study that we still are confirming. So my collaborators here at Columbia did the molecular dynamics analysis, and at least in this analysis, what was interesting is that 1961 really stabilizes the protein, because all other variants destabilize the protein, and that's kind of obvious. But in this case, it looks like it stabilizes. So what does this mean? We don't know. We still have to figure out if that's the case in other experiments. But, you know, this is one way to suggest that this specific variant, for whatever the reason, has a different effect on the protein than all other variants. Now, as I mentioned, we are working now on a lot of modifiers, and I will show you again on the example of 1961. The cis modifiers, we have found a lot of them. So these cis modifiers are variants that are on the same ABCA4 allele that the disease-causing variant, but they do not cause the disease on their own. Yeah, so they enhance the penetrance and expressivity. And here is this paradox of 1961, because people were saying, well, it is so frequent, so where are the homozygotes? Yeah, so there should be homozygotes. We have quite a number of homozygotes in our cohorts here. But just the fact that there are no homozygotes, people use that to discredit, you know, that this variant is disease-causing. And it is always penetrant in compound heterozygosity. And I always said that, you know, before making statements, you have to figure that out. So what we figured out is that if a patient or just a person is homozygous for 1961, the disease will develop if at least one allele has a modifier. And the most prominent modifier we found is this deep intronic variant that kind of raises the threshold, or let's say probably the other way, lowers the threshold of penetrance. Yeah, so if you are homozygous for 1961, and then there are, of course, different haplotypes in this locus, you will not develop the disease, at least in any reasonable time, maybe, you know, very late in life when it's confused with AMD. But if you have this variant minus 784 on one allele, you get the disease, not early, but you get this bullseye phenotype. And again, you can see that the penetrance is affected, but also you can see that disease expression is affected. Yeah, so that the cases with this modifier have a more severe phenotype, although the basic phenotype still stays the same. It's still the bullseye. It could be bigger or smaller, but it's the bullseye. And another good example here is how modifiers affect the disease is the compound heads. Yeah, that the other variant is this severe 1380 variant in both of these cases. This patient doesn't have a modifier on 1961. This does, and you can see how this affects the disease expression. So this concept of cis modifiers, and we have a bunch of them and on other alleles, and I will not be able to get into all of that, is a very important thing to consider in diagnosis of Stargardt disease, ABCA4 disease, and in prognosis of that. So again, coming back that, classical Stargardt disease was always considered, and this is actually the actual figure from Carl Stargardt's paper, where there are flecks and then there is, the macular degeneration, and it was always said that this is classical Stargardt, this is how it looks like, and the onset is childhood or young adult. Now, again, about 5 years ago, we brought in this hypomorph definition, showing that a huge fraction of ABCA4 disease actually is late onset. And it all started with this variant 1868. This variant is even more frequent in the general population than 1961. It's 7% in European population. And of course, automatically it was considered, benign because it can't be disease causing. But when we did this more detailed analysis and separate the cohort into 1 mutation cases, 2 mutation, 0 mutation cases, we noticed a couple of things, that if the allele on the, if a variant in the opposite allele was deleterious, then 1868 was penetrant. So this is the condition, and I'll go back. This is the condition when this variant becomes penetrant and does not need any other cis modifiers. And you can see there are a couple of interesting things. The disease onset is late. And why it is late? Because most of these cases have foveal sparing. Yeah, so they have still 20/20 vision. You can see that you can have a major GA, geographic atrophy, but if the fovea is still quite intact, and here it's on autofluorescence and OCT, about 90% of these cases have a foveal sparing, and then in their 50s, 60s, sometimes quickly they lose it, and then that's why people are asking, I was fine, what happened? So this is this fraction of late onset Stargardts. And it, of course, solved half of the 1 mutation cases. And what was also important, that it differentiated Stargardt from A and B. And again, when people ask that, I was wrong when I said that ABCA4 is associated with A and D. No, I was right. And still, you have to look at these things a little bit philosophically. What is late onset Stargardt and what is A and D? They are both age-related macular degenerations. Yeah, so if you look at it this way, that's that. And I can tell that if we take out all Stargardt cases from our A and D cohort, so the ABCA4 variation is still elevated, specifically this 1961 variant and so on. So there is still some, but again, now it gives you a pretty good idea how you separate them. And you can see, if you look at these 2 cases, these are 8 years time lapse, and on the left there is an early onset severe case, and on the right there is the 1868 late onset case. And And you can really see how different is the disease, how different is the progression. You can appreciate the foveal sparing here, and so on and so forth. Now, the other variant that we figured out was the 863 variant. Again, it is a very, unclear variant because it was considered, it is frequent. It is about 1 to 2% in Western Europe, suggested to be mild. Functional studies show it is severe. And it was suggested that it should have the same configuration, like we now know about 1868. But when we looked at our database, the other allele on the other side of 863 was not always deleterious. And so then we figured out that actually the 863 is pretty much always, there are a few cases, a few exceptions, but always insists with 1868. And so when these, and as I said, that this is again forms kind of a complex allele, but not in the real sense. If these are on the same allele, this allele becomes fully penetrant and is sometimes even severe. So again, the question, what is a modifier? What is the hypomorph or a mutation? Here it shows that 1868 doesn't need 863 to be pathogenic. 863 does. So there could be, again, exceptions. There is always an exception, but this is how these things currently are. Then we looked a little bit at trans-modifiers, and I'll run really quickly about this. Again, this study was published recently. So as you remember, I showed you this, yeah, that, there are phenocopies quite often. And so we used this information. We looked at the 7 genes that cause similar phenotypes than ABCA4 and asked the questions that are rare variants in those or common variants in those. Do they modify ABCA4 disease? And again, interesting findings where we did that in discovery cohorts and replication cohorts. So it really showed quite well two things. One is that rare variants in ROM1 are associated with are modifying ABCA4 disease because they are about 4 times higher, in cases than in controls. No other gene, rare variants in no other gene, including PRPH2, were associated with ABCA4 disease. But very interestingly, so there is this haplotype, on the C terminus of PRPH2. These 3 variants form a haplotype that is in about 10-20% of all population. And so when we used one of these variants as the haplotype tagging variant and looked at the association, we found kind of interestingly that, if we looked at the entire cohort, there was an association. And if we looked at 1868 separately, there was a reverse association. And so this just shows the numbers. The numbers are quite impressive, both in discovery, in replication, and in combined cohorts. 1961 was not associated subgroup with that. And so again, these are the first trans-modifiers for ABCA4 disease. Here, the influence is, not as clear as with cis modifiers. We could not determine, of course, the numbers were relatively small, especially for rare variants. We couldn't determine, phenotypic differences. But the common haplotype is really quite interesting because This we call it EKG, yeah, so by amino acids, have been shown that is associated with more severe disease by study in some years ago, and also it shows that it affects protein folding. So it could be, and that's where you see that the strongest association is with these hypomorphs, that it is an additional insult that somehow enhances the disease penetrance in that subgroup. And the last part of the study that is performed by Winston, that is also published, is what is the precision ophthalmology that I call it now. So what Winston did, because even 5-6 years ago, yeah, when people said, okay, so I have these two alleles in ABCA4, what is my outlook? What is my prognosis? I said that, you know, except for knockouts, where we could say that, you know, the disease starts early and progresses fast, you know, it was a mess. Yeah, so you couldn't say anything. Now, what Winston did is that first we defined the 4 disease progression groups, yeah, so macular, extramacular, so-called critical stage, and then of course advanced. And you know, this graph we already published at this review, in that review that I mentioned, that shows that if you have a hypomorph, you know, 2 of them are here described, your onset is late, yeah, it's, you know, 30s, 40s, and you will end up with this extramacular phenotype, but you will not develop pretty much into critical stage, nothing to talk about advanced. And then here is this famous 1961. The same thing here, the onset is about 20 years, so it is not that late, and you get this bullseye relatively quickly, but again, you will not get into the critical blowout stage. Now, when you have so-called classical disease, it starts, you know, yes, teens and progresses relatively quickly. And then if you are a knockout, unfortunately, the prognosis is very poor. It starts in the first decade, and by, you know, mid-20s, your vision is pretty much gone. So what Winston looked at were the specific cohort that were over 50 years of age, because every study that has looked into progression and Stargardts and this and that has looked at pretty much the patient at the time of diagnosis, or maybe sometimes does some follow-up. Here, the idea is that if you are over 50, your disease is pretty much where it will be, you know, in terms of Stargardt, ABCA4, that you are likely not really progress much further. And so the step 1, of course, is to look at these prognoses. Step 2 is classify mutations by severity. And again, the paper is out, so you can go and take a look specifically how we did that and what were our assumptions. And then you really do the matrix. Yeah, you integrate the genetics with clinical imaging observations, and that gives you the matrix. And the idea is to give this matrix to clinicians, to genetic counselors, to people who are not intimately familiar with Stargardt disease or ABCA4 disease in general, that after the clinical workup and gene screening, you can put the data into this matrix. And so then you can say that if you have 1961, you will be, doesn't matter what is the other allele. If it matters a little bit but it will never develop into this critical stage, point of no return, that my colleague Steve Tsang likes to say. And with hypomorphs, it is a little different, because some of them, as I said, very late can develop into more severe disease. But again, most of it will be in the prognostic group 2. Now, of course, if you are a knockout, then unfortunately the disease outcome is really poor. The prognosis is poor, and these are the cases where any kind of treatment option that becomes available, any clinical trial should be hitting these, because by the time they're in their 20s, it'll be too late, and you have to just regrow the retina, and that would be the only thing to do. So this gives you, the general idea why we're talking about precision ophthalmology in ABCA4 case, and this is the most typical case, I think, in Mendelian monogenic retinal diseases, that we went from clinical medicine to genetic medicine, but now we really can do more of a precision ophthalmology. Of course, this is a moving target. This was analysis of 100 cases there. Anytime new data comes in, should be included, because only large numbers will give you the more specific and better answer. So the ABCA4 locus, I probably showed you, is a very complicated locus, and only comprehensive and integrated clinical and genetic research will result in proper diagnosis, prognosis, and treatment. And I'll close with this slide, because I noticed Kapil was on this. So when I saw this headline, I said, well, I thought I've seen everything in my life about ABCA4 disease. Yeah, I said, what? But I'll leave it at that at this point. Thank you very much for your attention.
Thank you very much, Rando. Any questions you have, you don't need to write it. Let me know you have one. We will have final comments coming from Dr. Debashi Mustafi. Thank you for that. So let's start with Caroline.
Hi, Dr. Allikmets. Thank you for the you showed the genetic variants associated with ABCA4 diseases. Do you think that the animal models we have available would reflect a good starting point to study therapeutics related to Stargardt?
Can you put the screen up? Okay, now I can do it myself, I guess. So who asked the question?
Caroline from the room.
Okay. And the question was about the animal models we currently have. Is that correct?
Yeah, that's it.
That's a very interesting and important question, because the only model now, maybe there are a couple of others, but is really the mouse. Yeah? And the mouse is not a good model for Stargardts, and we've seen that, but that was what we had. Now I've seen there is a rat, but I don't think that makes that big a difference. And also the zebrafish that was presented at ARVO. I don't know if it's published or not. So again, if you look at the mouse, even the knockout mouse has high lipofuscin accumulation, but that's about it. There is a outer nuclear layer thinning probably, but really the well quantifiable criterion when you use this mouse for let's say trying to find treatment for Stargardt disease is the lipofuscin accumulation. Now this said, many variants, as I showed, do not accumulate highly like diffusion, and I can tell that specifically for 1961 also in the mouse. It is pretty much like the wild type. So it is a problem. We have to deal with this, as it is. And when I was talking today about, different studies, whether these are in vitro or in, kind of in vivo, we are all trying. For example, there is even retinal organoids are not a very good model for ABC4 disease, for the simple fact that, there is no visual cycle going on, where ABC4 really works. You can look at some structural changes, you can look at some things, but these are not good, as are not good the RPE cell cultures. So it is a problem. It's not that people don't know what they're doing or they're not trying, but so while I know that there are now Stargardt dogs available, so that is a little bit closer, but again, what is the number of those animals? So that's another good question. So the good model for macular dystrophy, like ABC4 disease is still not available in my mind, but we work with what we have.
Humans. Rando, so following that question, is it possible that because rodents, particularly mice, have almost 10 times higher metabolism of drugs, they could metabolize the aldehyde, that's the reason why those mice display the phenotype that we will see this in humans, is simply that the reduction power, oxidation power is so much stronger as compared to humans. And that's the reason why those animal models, we make, as you know, double knock-in, and you make knock-in and knockouts. They really do not recapitulate the human condition. It's not juvenile observation of changes for sure in those mice. We can argue whether that it is later on in life of those mice, but for the first year, there's really no major phenotype, as you mentioned, only the accumulation of A2E and other adducts.
Yeah, I think you're probably correct. That's probably one of the reasons, but I think the first and major reason, and everybody acknowledges that, is the lack of fovea, the very small number of cones in mice, and specifically if you have, because now whichever variants these are in ABCA4, they do affect cones in the first place. So it is not as drastic as 1961 that really goes after them for whatever the mechanism, but this is how the disease is. And so always, I remember when Peter Gouras, when he was still here and alive, he was also asking why macula. So probably the answer is cones, because it is either Stargardt or cone rod, or it is not rod cone, and so on. But yeah, you're right, metabolism is the other thing, and that's why it is very important to assess your result in any model system, whether it's a mouse or another animal or a cell culture or a in vitro with a grain of salt, because this is not the real situation, to be absolutely clear.
Let's go to Crystal.
Yes, hi there. Thank you so much. That was a wonderful talk. So I was just curious for the G1961E mutation. You had showed the ABCA4 was localizing where it should in the outer segments, but I was wondering if you happen to look at the retinal pigmented epithelial cells as well, and if you did, if you saw any abnormalities there.
No, I didn't. So see anything here, because now, if you do the cross section, you have them there. So that's another long story, and probably we will not be able to discuss this, but let me put it this way: I have not seen any reasonable expression of ABCA4 in the RPE. and certainly not at the level where it could be considered functional. So in vivo, again, if you make a system, it could work, because that's another artificial thing. Because when we did the first gene therapy for ABCA4 in lentivirus 15 years ago, that somehow fizzled. We had this discussion with Janet Sparrow, with whom we did that, is that when we injected, transduced the newborn mice with lenti, because in adult mice, lenti doesn't go to the photoreceptors much at all. So we obtained about 20% or so efficacy, what is quite low. However, the effect based on, again, lipofuscin measurements was profound. And so then we were thinking that maybe if you put, ABCA4 in the RPE, it may have something to do with that effect. So I'm not saying that it cannot work there at all, but I'm just saying that physiologically, I don't think that's really the case.
Ben?
Great talk, Rando. Thank you. I was wondering, given that people with the 1961 mutation don't have accumulations of lipofuscin and A2E, do you think that therapies targeting vitamin A or modulating vitamin A will be less effective for those people?
Yes. So that's another point I did not cover today, and that is a very important point. And we still actually have, I think, the last year of the grant from FFB, where we're looking specifically, how to select Stargardt patients for clinical trials. And because if you are targeting, you have to, depending on what the modality is, whether it's, the visual cycle thing or vitamin A access or gene therapy or whatever, you really have to pick patients that could have some effect, because picking, these cases that accumulate little lipofuscin will not show any effect, even if there was, because that's kind of very simple. If there was, they don't show it, because Stargardt disease mostly is slowly progressive disease, so we know that. And so actually doing a clinical trial in Stargardt disease is much more complicated than one would think. So that's one thing, because it's slow, and you can do a 10-year trial. You have to do it like 2 years or so. The other thing is that the only accepted by FDA criterion is this expansion of the geographic atrophy, and a well measurable geographic atrophy happens in less than half, if not more, of Stargardt patients. So you cannot really use patients that don't have that feature. And so that's why these trials recently that were published or talked about use those late onset cases where you have this well demarcated GA, and that you can really measure. But then again, it is slow. So it is very, very difficult to do the correct clinical trial for Stargardt disease. Again, pending, of course, the modality that you are using.
Great, thank you.
I see there.
Okay, so the next
Vladimir is
question.
there. Let me ask a question. So we have these mice that we discussed a few years ago, GNAT1 double. You want to do the measurements?
Hey, Rando, yeah, of course. I'm happy to follow up on that. And we can talk offline. We were actually talking with Sasha from my lab, and he was asking me whatever happened with those mice. So yes, thank you.
We've been making them. They are, it's not, it takes time.
Okay, we can certainly follow up, and I'm happy to collaborate with you on that. Maybe a naive and kind of question that reflects my ignorance, but can you comment on the mechanism by which, obviously you have this diverse group of mutations. And what is the current thinking of the mechanism by which all of them lead degeneration with very different clinical manifestation, different rates? What is the current kind of understanding in the field about the link between mutations and the actual degeneration?
No, it really depends, I mean, this is kind of standard genetics question. Yeah. So how a variant affects the protein. So in ABCA4, you have, all these different categories. Yeah. So, I mean, severe variants, it's clear. Yeah. If it's a stop mutation or a, frame shift, variant. Splice, yeah, plus minus one, and so on. So there is just no protein. So that's a very simple thing. Now, I have been discussing this quite a bit with my good friend Franz Kremers, with whom we lately argue a lot because we probably are just old. But we do agree on some things. So, for example, he's studying a lot of those deep intronic variants. So deep intronic variant results in mostly in pseudogene insertion or sort of pseudo-exon insertion, or then, the exon spliced out. And pretty much that takes care of, the messenger, the protein on that allele. However, they all have different rates, what they do. Yeah. So some take it 100%, some take it 50%. And so what we always have been discussing, so what is the residual ABCA4 activity that is necessary for the disease not to develop? Yeah. So this has always been this thing that it is clear that, if you are missing half, you're fine. Yeah. So, I mean, no, there are these, again, controversial things that carriers have phenotype late in life, and they might, but this is not the major issue. But basically, if you have half the protein, you're good. Now, the question is, how much less is bad? Yeah. So we have put the number currently at 25% of the other allele. So if you have, 75% of one allele working, so that should do the trick. And so that is also very promising for clinical trials. Yeah. So you don't need to restore Stargardt activity to 100%, or, so you just add a bit, and, oftentimes it does the trick. So, but again, different mutations, they influence the protein differently. Yeah. Some, just don't make it. Some influence the assembly, some influence the trafficking, a lot of them. Yeah. So because they get stuck in the ER, and that has been shown. And so there are many, many ways. And then there are those like 1961 that everything seems fine, but it's not working. So, and specifically in cones, yeah, for some reason, and why, we don't know, or primarily. So that's, these things. But since, this vast number of variants, yeah, over 2,000 already, so shifting through them is very difficult, yeah, because most variants are rare, very rare. They haven't been seen. Yeah? So, and if you expand even out of European population, then it gets totally messy. So it takes a lot of work to figure it out, and there are just objective problems, yeah, with this. But we try.
Right, so let's go to last two questions, John and Sam, and then final comments. We'll switch to Don Sweater-Zak.
Hello, Dr. Allikmets. Thank you for the amazing talk. As you know, there's lots of not lots known about ABCA4 and its role in the rods, in the cellular architecture and the biochemistry. And you nicely show this macular degenerative phenotype with the ABCA4 diseases. I was wondering if you can elaborate a bit more on the ABCA4 role potentially in cones, and whether this is ABCA4 disease is more of a rod specific thing that eventually affects and starves out the cones, or if you had any thoughts on that.
If I'd known the answer, I mean, I wouldn't give today's talk. So, So this is what we all try to figure out, yeah. So and again, it goes back to the same 1961. No, Stargardt disease, ABCA4 disease, is the disease of cones primarily. So that is clear. Yeah, so it's a macular dystrophy. It is primarily cones. It also affects rods later. Yeah, so that's how it is. Now, there has always been this question, you know, since it is in the outer segment disc rims. Yeah, so the question is, okay, in rods, it is closed environment. In cones, it is different. So what happens there? Again, I don't know the answer. And that's what we really try to figure out, you know. So I think that something that's going on with 1961 has to do some specific issue with cones, but it is a cone disease primarily. It is a macular dystrophy. Now the mechanism, we don't know. We still have to figure that out.
Hey, Sam.
Hi, Dr. Allikmets. Thank you for your talk. I'm a MD PhD student in Chris's lab, and I'm kind of interested in what you think about the window of intervention for these Stargardt patients. If they present in clinic, is it already almost too late for intervention and we need to find and treat them sooner? Or do you think that they're still treatable when they come into clinic for their diagnosis?
Well, this again depends on what stage of the disease they have when they come to clinic. And then based on that, of course, you know, there could be different intervention possibilities. Again, there are, I already said, yeah, there are so many of these different clinical trials that are going on trying to treat one way or the other. I'm still a big proponent of gene therapy, specifically because this is a recessive disease and you have to add some function, and that does it. But then again, we understand clearly that it only works if you have cells left, yeah, specifically photoreceptor, maybe RP, to put the gene in. So that is a good question, and that I pretty much covered that before when I was discussing different clinical trials, different modalities. Those with most severe disease, unfortunately, and usually, you know, by mid-20s, they have really lost most of the cells, and it's really, it doesn't really matter, you know, what modality you use. But I am encouraging, you know, everybody to, you know, take advantage with whatever there is available. Yeah, so I'm not a big fan of the, you know, deuterated vitamin A thing, but if that's the only thing there, why not? You can try. I mean, it doesn't hurt, I guess, you know. So, and I think that this category of drugs of vitamin A or the visual cycle modulators, whichever way it's done, should have some effect. Yeah, so I would expect that. But again, you know, they have an effect in a certain class of patients or certain subgroup of patients. So again, it's quite complicated. So again, in terms of when they come to the clinic, you have to see, you know, what is their genetics, what is their current stage of the disease, and then, you know, look and advise, you know, what they can do specifically, you know, with clinical trials that are currently ongoing. Yeah, so it's I don't think, I mean, I'm frustrated myself, and patients write to me, although I am not the clinician, that, well, every time we hear that in 5-10 years there will be, you know, a treatment, I say, well, I was hoping so too. And it's frustratingly slow is how all these modalities move through clinical trials. And there are many, many reasons for that, as I already mentioned. So I would tell patients not to lose hope and still be, you know, optimistic. However, you know, you have to give it again with certain more specifics in terms of what to expect.
Tom, your final comments and final questions.
I forgot my sweater. But Rando, thanks very much. That's really an elegant summary of, you know, literally decades of work. So thank you. I have a couple of questions. One is, given that the visual cycle is partly extracellular, have you or anybody made chimeric mutant mice where you have cells which have the mutation next to cells that don't, and shown that the wild type cells are affected similarly to ones carrying the mutation?
No, we have not, and I am not aware of anybody.
Is that worth trying? Would that teach us anything, do you think?
Yeah, again, I don't know, because I mean, everything in Stargardt is empirical. And that's what I always say, that, you know, you have to figure out what it is, then you can, you know, say, because my opinion is one thing. I
Okay.
like to base this on fact that I currently know, but this fact can change.
But Randall, you like your opinion, admit it.
No, I mean, I used to when I was much younger, you know. And when we were talking about AMD, now I'm really, you know.
All right,
Okay.
with that, let's join me in thanking Rando for.
Thank you.
Rando, you have changed.
So Randall, this is offline since we're already over, but the other question I wanted to ask you is about the retinal organoids that you mentioned. What kind of phenotype do you see in the organoids?